Let's look at the modes of regulation of enzymes, or in other words, how do we control enzyme activity, aside from what we have seen, the allosteric control. Another mode of enzyme regulation is phosphorylation. Recall from your organic chemistry that an alcohol plus an acid produces an ester. Reminder, ester is a high energy bond and it undergoes hydrolysis with a release of energy. One thing that you may not have been introduced in your organic chemistry is that instead of a carboxylic acid, the acid can be a phosphoric acid. So an alcohol plus a phosphoric acid produces a phosphoester. This reaction is also known as a phosphorylation. You are phosphorylating the alcohol. Which amino acids can undergo phosphorylation? Can you recall? That's right. Serine has an alcohol group. Threonine has an alcohol group. Tyrosine is a phenol, so it can also be considered as an alcohol. So these amino acids can undergo phosphorylation, and if the source of the phosphate group is ATP or similar compounds, then they're catalyzed by enzymes called kinases. Okay, so visually speaking, we have, say, serine with ATP. Okay, so ATP, the enzyme will be a kinase. So the rest of the protein has been, the serine residue has been phosphorylated to form ATP. Now, what is the effect of this one? Okay, so recall again your tertiary structure. What intermolecular forces of attraction do you think serine would participate in? Would it be the same if instead of an alcohol group, you now have a phosphorylated group? No? So recall, what is this? Alcohol. So most likely it's involved in hydrogen bonding, either as a hydrogen donor or a hydrogen acceptor. Is that possible here? What about the space requirement? Okay, will it change? So do you think the conformation of that protein will change? Other residues, threonine and tyrosine. Okay, so you have the threonine residue and then the phosphorylated threonine residue, tyrosine residue, phosphorylated tyrosine residue. Okay, so let's look at one of the effects of phosphorylation as a mode of enzyme regulation. One enzyme is the sodium-potassium pump. Why is it called a pump? Now, what do you do when you're pumping? You're moving something against a concentration gradient. Okay? So normal conditions in the cell, sodium ion concentration inside the cell is less than the sodium ion concentration outside. Potassium ion concentration inside the cell is normally higher than potassium ion concentration to the outside. What is the tendency? The tendency is for the sodium ion concentration inside and outside the cell to equal each other. The potassium ions inside the cell, concentration inside the cell equal the concentration of potassium ions outside the cell. But you don't want this. Once it reaches equilibrium, the cell has long been dead. Okay, so what does the sodium-potassium pump do? It should counter the natural tendency of the sodium ions to go out, to go into the cell, and for the potassium ions to go out of the cell. Okay, so there is constant pumping of sodium ions to the outside of the cell and constant pumping of potassium ions into the cell. So let's look at what happens. So you have the enzyme with ATP. It's now phosphorylated. a unit, aspartate 369. Again, what does that mean? It means that the 369th residue in this enzyme is aspartic acid or aspartate in the normal sense for the cell. Okay, so ATP converted to ATP, one of the phosphate groups from ATP is now bound to the enzyme, again, specifically aspartate 369. You now have a conformational change. Okay, so that's the answer to the question earlier. There will be conformational change because of the change of the addition of the phosphate group. But this is conducive to, again, picking up 3 sodium ions from the inside of the cell, producing also another conformational change because it has now picked up sodium ions. But it's also conducive to pumping these 3 sodium ions from the inside to the outside. So once again, you have this enzyme that has just undergone conformational change, and it's now susceptible to hydrolysis. So the phosphate that was added is now hydrolyzed by water, okay, it resulting in a dephosphorylation of the same aspartate 369 and producing another conformational change, this time picking up two potassium ions from the outside and bringing them or pumping them into the inside of the cell. Hence, keeping this ratio of sodium ions and potassium ions that keeps the cell alive. Another example is glycogen phosphorylase. This is interesting because there are many ways to control the activity of the phosphorylase. And however, to complicate our lives a little bit, okay, glycogen phosphorylase is very common, so it's usually abbreviated to just phosphorylase. And it has two forms, phosphorylase B and phosphorylase A. Phosphorylase A is phosphorylated. Okay, so please keep track of all these terms. Both of them can exist in the T form, meaning they are in the inactive site. We just came from talking about covalent control, which is phosphorylation. So phosphorylase B can be phosphorylated by the enzyme phosphorylase kinase. It is dephosphorylated by the enzyme phosphoprotein phosphatase. Okay? So again, it changes conformation. So there again. So far, glycogen phosphorylase can be controlled covalently by phosphorylation and dephosphorylation. However, the two forms of the glycogen phosphorylase, okay, can also be controlled allosterically. Phosphorylase B is activated by the presence of AMP into the R state. Phosphorylase B is inhibited by higher concentrations of ATP, the presence of glucose 6-phosphate, glucose, and caffeine. These are allosteric effectors to phosphorylase B. Phosphorylase A, on the other hand, is not activated by allosteric effectors. However, the R state of phosphorylase B is deactivated or inactivated by the presence of glucose and caffeine. Interesting, right? Another method of controlling enzymes is through the zymogens. Okay. So what are zymogens? Okay. So some enzymes are synthesized in the cell in the inactive state. How are these proteins converted into the active site? It involves the breaking of some peptide bonds, which are covalent bonds, and sometimes also the removal of some residues. So these types of proteins, enzymes, are called zymogens. For example, you might have already heard of chymotrypsinogen. This is the inactive form. Breaking some peptide bonds converts it into chymotrypsin, which is now the active form. Trypsinogen into trypsin, prothrombin into thrombin. Examples of zymogens are the proteolytic enzymes trypsin and chymotrypsin. More specifically, we will illustrate the chymotrypsin as zymogen. Chymotrypsinogen is synthesized in the pancreas first as an inactive zymogen. Notice that there are now disulfide linkages shown here in the yellow. Okay, and the whole polypeptide chain is from amino acid residue 1 to amino acid residue 245. This is still in the inactive form. Existing trypsin is needed to cleave the peptide bond of arginine 15. So this one is now cut. However, notice again that there are existing disulfide linkages, so the polypeptide chain does not fall apart into different separate strands. Now, this is pi chymotrypsin, which is now active. It is able to self-digest, meaning pi chymotrypsin can act on another pi chymotrypsin molecule, and it cleaves the peptide bonds between amino acid residue number 13 and 14, number 146 and 147, and 148 and 149. So these dipeptides have been removed. Once again, I'd like to emphasize that there are existing disulfide linkages that holds the whole polypeptide chain still as one piece. Now in this form, this is now the alpha chymotrypsin. This is now the fully active enzyme and is now able to hydrolyze proteins that's in our digestive tract. Another example involved in blood clotting is fibrinogen in the inactive form. And when it's necessary, for example, you have cuts, then the fibrinogen is hydrolyzed into fibrin. It is now active. Another example is involved in blood clotting. Fibrinogen uh, circulates in our system, but then is only converted into fibrin by, again, cleaving some of the peptide bonds when it's needed for us to have blood clots.